Hello, we shall discuss on the next plant growth regulator, that is ethylene. Ethylene is a gaseous phytohormone synthesized in large amount by the tissues that is undergoing senescence and ripening of fruits. Ethylene is highly effective in fruit ripening and it enhances the respiration rate during ripening of fruits. So this rise in the rate of the respiration during fruit ripening is called as the respiratory climactic. Ethylene breaks seed and bud dormancy and initiates germination of seeds in peanuts and even sprouting of potato tubers. It also promotes rapid internode elongation in deep water rice seed plants, and this helps the upper parts of the shoots of the rice to remain above the water. Ethylene also promotes the root growth and root hair formation, thus helping the plants in increasing the surface area of absorption. Ethylene is used to initiate flowering and for synchronizing the fruits set in crops like pineapples, and also induces flowering in mangoes. The most widely used compound of ethylene is the ethaphone. Ethaphone is an aqueous solution containing ethylene, and it releases this ethaphone releases ethylene slowly, and this ethaphone hastens or increases the fruit ripening in tomatoes, apples, and accelerates the abscissions in flowers and fruits of plants like cotton, cherry, walnut. Ethylene or ethaphone also promotes female flowers in cucumber, thereby increasing the yield of this plant. Now the next type of phytohormone is the abscisic acid. Abscisic acid acts as the plant growth inhibitor and also inhibit plant metabolism. The abscisic acid inhibits seed germination and stimulates the closure of stomata in the epidermis, and it increases the tolerance of plants to various kind of stress. Stress here refers to external environmental factors or both internal environmental factors. Some of the external environmental stress includes temperature, heat, cold, etc. So by closing the stomata in the epidermis of the cells, the abscisic acid help the plants to increase it increases the tolerance to the environmental stress. So because of these reasons, abscisic acid is also called as the stress hormone. This abscisic acid also helps the seeds to withstand desiccation and other unfavorable environmental factors for growth. Desiccation here refers to drying of seeds or losing excessive water. And in most cases, abscisic acid also acts as antagonist to GA. Here GA refers to gibberellic acid. So the action of abscisic acid is quite opposite. That means is in opposite to that of the gibberellic acid. If gibberellic acid stimulates growth, then abscisic acid will inhibit growth. So that is the meaning of antagonistic effect. Now the last phytohormones is the cytokinins. Cytokinins are substances that are believed to enhance the process of cytokinesis. The discovery of this cytokinesis started with two persons, that is Scoot and Miller. This person observed that cells of internodal regions of the stem of tobacco plants shows proliferation, that is cell division. The stem of tobacco plants shows cell division only when if auxins, there is a phytohormone called as the auxin, and other substances like extract of coconut milk, extract of yeast, extract of DNA were added to the growth medium that is containing tobacco stem. So that means in this substance, that is the extract of coconut milk, yeast, and even the DNA, there is a particular substance which together with auxin helped to induce cell division. So this substance were later found to be as kinetin. So Scoot and Miller discovered the kinetin, and later on this kinetin was named as cytokinins since it induces or help in cell division. Cytokinesis refers to cytokinins here refers to the substance which induces cytokinesis. Cytokinesis means, cyto means cell, kinesis means division. So basing on its action in proliferating the cell division or increasing cell division, this active substance was known as cytokinins. Now let's discuss on the functions of cytokinins. Cytokinins promote cell division, and it also promotes cell enlargement. That means increasing in size of the cell, and also cell differentiation. Cell differentiation here refers to the helping the cell to become specialized to perform a specific functions. These cytokinins also help in the formation of adventitious roots. Adventitious roots are those roots which originate from any part of the plant except the radicle. These cytokinins also help in the leaf formation and leaf enlargement, and it break the seed dormancy. Cytokinins also delay senescence. Senescence here refers to biological aging, and it promotes flowering in some plants. And these cytokinins also help in formation of intervesicular cambium. This intervesicular cambium are those cambium which are formed between the intravesicular cambium during the secondary growth. So this is all about the growth regulators or the phytohormones. Now we shall discuss on the seed dormancy. Seed dormancy means the failure of the seed to germinate even under favorable conditions. There are various reasons which induces seed dormancy. So let us discuss some of the few reasons. The first reason that may cause seed dormancy is, one is that due to impermeability of seed coat. If the seed coat is impermeable here refers to not allowing water or any other substance to enter the seed. So if the seed coat is impermeable to water, then the enzymes and the hormones cannot be produced. So this may result in the inability of the seed germination. The second reason is underdeveloped embryo. That means this embryo have not reached a stage where it is capable of growing. Third is that high levels of inhibitors. Here there are different inhibitors which inhibit the development of seed, growth and development of seed. However, we are referring mainly the abscisic acid. The fourth is the hard seed coat, and the fifth is the requirement of low temperature or vernalization. So these are some reasons that may induces seed dormancy. Now let's discuss on how this sort seed dormancy can be broken or seed dormancy can be overcome. The first is that treating the seed with acids to soften the seed coat in order to break the hard seed coat. So when the seeds are treated with acids, this soften the seed coat, and when the seed coat is softened, it allows the germination of seeds. So this treatment of seeds with chemicals or acid is called as chemical scarification. The second is mechanically, the seed coat can also be broken by scrubbing on the hard surface. So this method of breaking down of seed coat mechanically is called as mechanical scarification. The third method can be through natural means. Naturally, the seed coat can be broken by the enzymes that are present inside the seed itself. The fourth is that treatment of seed with growth hormones like auxins, gibberellin, cytokinins. So sometimes the seed does not get a perfect temperature or perfect environment, or sometimes the seed itself cannot germinate. So by providing growth regulators like auxins, gibberellin, and cytokinins, the seeds starts to germinate. The fifth methods to break this seed dormancy is in case of seed dormancy is caused by a high level of inhibitors. That means because of the presence of inhibitors like abscisic acid. So in this case, treatment of seed with oxidant can oxidize these inhibitors, and once the oxidation of these inhibitors takes place, favorable environment, once this oxidation takes place, the inhibitors no more remain in the seed coat since they are being removed through oxidation. Therefore, Once this inhibitor is being removed through oxidation, the seeds can be allowed to germinate. So these are some of the methods to break or overcome seed dormancy.